I'm very excited to be back here at Bar Ilan for the second international conference on halacha and bioethics. Today, you will be exposed to the most current, cutting-edge biotechnologies in the area of genetics and reproductive medicine. These technologies are here, and they'll have a really pronounced effect on your lives, on the lives of your children, and the lives of your grandchildren. And in this multidisciplinary conference, we have really recruited some of the most renowned experts in the areas of science, halacha, law, bioethics, and psychology to analyze how these new technologies will impact society. In addition, we today will present to you some of the experts in halacha and bioethics to try to attempt to resolve and manage the emerging ethical issues arising from these biotechnologies. The ability of our halachic authorities to confront and to deliberate these bioethical challenges is really remarkable. I first of all want to thank the organizing committee for this conference. First to Professor Uri Nir, the Dean of Life Sciences at Bar Ilan, and to Rabbi Daniel Sperber, who is the president of the Institute of Advanced Torah Studies at Bar Ilan, to Rabbi Shabtai Rappaport, who's the Rosh Beis Medrash of the Advanced Torah Institute at Bar Ilan, and finally to Rabbi Altshul, who is the director of the Jesselyn Institute of Advanced Torah Studies. Rabbi Altshul and I go back many, many years, and it's really the first time we've worked together, and it's been really a pleasure, and I think Bar Ilan should be very privileged and appreciative of all his efforts to have such a talented person here at Bar Ilan. I also want to thank their staff. I don't know all the staff, but just highlight two people. Kobe, who was part of the 2005 conference that we held here, and has always been a catalyst to ensure that these conferences proceed smoothly. And also to Diana, who is Rabbi Altshul's administrative assistant. Now, I want to take 2 minutes just to thank the sponsors who have provided both financial and intellectual support for this conference. From Columbia University, you will hear Dr. Ruth Fishbach, who is the director for the Center of Bioethics. She has been a mentor and a colleague to many of us at Columbia University. And we have Dr. Kenneth Prager, who's professor of clinical medicine and also chair of the Medical Ethics Committee. And I must tell you that he faces some of the most challenging medical ethics dilemmas that our hospital faces on a daily basis. And his ability and capacity to deal and to resolve these real-life situations is really a Kiddush Hashem for us. And I will introduce Eric Schoen later. And I want to thank the people from Albert Einstein College of Medicine, the Dean, Dr. Ed Burns, who's a longtime colleague and friend, and for sending Dr. Gil Atzmon, who's professor, Department of Medicine and Genetics, who published one of the seminal works on Jewish population genetics that was highlighted in the Jerusalem Post last Friday, and it's a paper that we all look forward to reading to in the next few days. I want to thank Touro College of Osteopathic Medicine, the Dean, Dr. Robert Goldberg, who has supported this conference from day one, and Dr. Gary Steinman, who's coming shortly. He's a professor and chair of biochemistry. He is a professor who integrates and translates both clinical research and academic skills in the area of reproductive medicine. I would like to thank Dr. Tzvi Weinreb, who is the Executive Vice President Emeritus of the Orthodox Union. He's a clinical psychologist and probably one of the leading halachic psychologists in the world. I want to give thanks to the Jewish press, to Jerry Wartsky, who is if you don't know him, he's a long life supporter of many Jewish causes in Israel. And I want to thank the Henry and Bertha and Edward Rothman Foundation, who has been generous in providing some support for this. And finally, I want to thank the International Academic Friends of Israel. IAFI, as it's called, is an organization that's devoted to promoting and supporting the free and open exchange of ideas and information in the international academic community. IAFI has been a major player in diffusing British, Norwegian, and even US university boycotts or attempts to boycotts against Israeli academics. And if you want more information, there should be some in your package. And they really have gone all out to try to limit the effect of these boycotts on Israeli academics. I want to give recognition to Nadav Kidron, who was part of the original conference and also a major player in making sure these conferences get off the ground. And finally, I have to say Achron Chaviv to Rabbi Moshe Tendler, who unfortunately could not come due to academic responsibilities at Yeshiva University. He is a firm supporter, both publicly and privately, in all these conferences, and a major player. So, as I speak all over the world, I'm constantly recognized and valued that our people, the Jewish nation, serves as the leader in promoting to all societies moral and ethical values, and especially in the area of medical ethics. This conference represents a small part of our directive and mission of Tikkun Olam, and it's something that can make us proud of our nation and of our people. And I think this is a very important outcome and message of this conference. And I'd like to introduce now to you Dr. Uri Nir.
Good morning. Thank you, John. I'm pleased to welcome our distinguished guests and partners from Columbia University. And the academical staff here from Bar Ilan, members of the מכון גבוה לתורה, guests from outside of the university, students, and with your permission, I'll address the audience now in a few sentences in Hebrew, and if somebody will find it important, it can be translated simultaneously. בסיפור הבריאה, בספר בראשית, כאשר הקדוש ברוך הוא יוצר את כל מרכיבי הבריאה, בכל שלב ושלב מתבונן הקדוש ברוך הוא על סופו של התהליך, ונאמר וירא כי טוב. הקדוש ברוך הוא מרגיש שהבריאה נעשתה כהלכתה, כהלכה, וירא כי טוב. היצור הוא היציר היחידי שבו לא נאמר וירא כי טוב, הוא ביצירת האדם. על האדם לא נאמר כי טוב. ואחד ההסברים הוא שנראה שבריאתו לא הושלמה. בריאתו של האדם לא הושלמה. האדם הולך ומשפר את עצמו. הולך ומעצב את עצמו. הולך ורוכש יכולות חדשות לכל אורך הדורות. וזה האתגר שעומד בפני האדם. הוא יצור שאינו מושלם. הוא יצור שצריך לשפר את עצמו, להרחיב את עצמו. והרכישת יכולות האלה בידי האדם במהלך הדורות גם מעמידה אותו בפני אתגרים חדשים. מה לעשות עם היכולות האלה? כיצד להתמודד עם הבריאה שמסביב? כיצד לגייס את עצמו לשיפור העולם, לתיקון הבריאה? כיצד לממש את חיסרתו מעט מאלוקים? כי בצלם אלוקים ברא אותם, את האדם ואת החווה. לגבי חלק מהאתגרים האלה אנחנו נדון בהם פה היום, בהקשר של גנטיקה והלכה, ואני חושב שאין מקום ראוי מבר אילן לדון בהקשרים אלה. לי עצמי כמדען שגדל בבר אילן, גם במוסדות בחוץ, והיום עוסק במחקר כאן, אני חושב שבר אילן מייצגת מוסד מאוד ייחודי שמקיים ערך של מורשת יהודית יחד עם מצוינות מחקרית, והחיבור הזה הוא במה נאותה ביותר לנושאים החשובים שבהם נדון היום, ואני מקווה שהמפגש הזה יפרה את כולנו, ירחיב את דעתנו, יעלה אולי שאלות חדשות, אבל גם נקודות חדשות לקידום הנושא החשוב הזה. אז ברוכים הבאים, and enjoy this, I hope, very stimulating conference.
Rabbi Professor Daniel Sperber.
ברשות הרבנים, אני ארשה לעצמי לדבר באנגלית לאלו שעדיין לא השלימו את לימוד השפה העברית. Ladies and gentlemen, I said that I would permit myself to speak in English for those of you who have not yet fully mastered the Hebrew language. Of course, I'd like to repeat the welcome and the thanks that were so eloquently expressed by Dr. John Loike. Our thanks go most specifically to him because it was his initiative 5 years ago and his indefatigable energy that brought about the first conference, and he's relentless. And he was the one who forced us willingly to have this second conference. In a sense, this is a sort of a continuation of the conference we had 5 years ago, and it's a real pleasure to see once again familiar faces and friendships that we formed 5 years ago. But in fact, it's also the culmination and the summit of a new program which we began a year ago and which has been carried on throughout the whole of this year. A program which we call Nitzotzot, Sparks. In this program, we have invited leading scholars from the different faculties of the university and from outside the university to present their latest thinking, their new theses, in whichever area of expertise that they have been involved in, whether it be economics, biophysics, sociology, psychology, and so forth, to present them to us and to have one of our senior students from the Institute of Higher Torah Studies over here to respond and to try to express to us what are the implications of these innovative thoughts within the field of halakha, of Jewish law. And then we have opened the panel, and our Rosh Beit Midrash, Rav Shabtai Rappaport, has given a short lecture explaining the complexities of the relationship between these new areas of discovery, of scientific discovery, and how it should relate to halachic thinking. The rationale behind this program was, and is, that every advance in all areas of science have their implications in halakha, in Jewish thought, and in Jewish ethical thinking. And therefore, in a sense, this conference constitutes the culmination of a year's activity in which we've tried to relate cutting-edge science, innovative discoveries, and their implications for halakha. Now, the reason that we have had this conference, or are having this conference, is not because we want to put ourselves on the map of academia. It's not because we want to appear in the middle pages of one or two newspapers. It's because we genuinely believe that the challenges of modern science are challenges which not only face Jewish people, not only face rabbis and halachists, but they're ethical challenges of a universal nature which challenge all of human society. In a sense, perhaps in a small sense, what we're trying to do is to input into these new ways of thinking how Jewish ethicists and halachists would attempt to brace themselves to these new challenges. The point of last year's conference and this year's conference is not merely to discuss contemporary situations in the fields of bioethics, but what the future potentially holds out for us in these areas. And in many cases, the future holds out wonderful opportunities, but also very frightening scenarios. And we have to prepare ourselves. We, as halachists, as rabbis, as ethicists, to learn what that future potential is in all its different manifestations, in order to attempt to grasp how we can go about dealing with these new problems which don't really have any precedence in our literature. And the way we do this is not to find a case which seems to be identical in rabbinic literature, but to try to plumb the depth of Jewish halachic thought in order to find what are the underlying principles involved, and then to try to form some sort of comparison with what is going on and what will go on in the next decade or so. So, in a sense, we feel that scientists who face these challenges themselves can learn much from us, can learn from halachists and ethicists, and we, rabbis and ethicists, have much to learn from them. And it is this dialogue that we are trying to constitute in these conferences. And perhaps, if even in a small way, we can contribute to the problems which are facing world society and which are of a world nature. Perhaps if we can contribute some insights into the way to tackle these problems, to face them, and perhaps to make hard decisions as to which areas to develop and which areas not to develop, or how to restrain ourselves in various areas and to develop other areas, perhaps in this way we can have some sort of input into this universal challenge. Thank you very much, and once again, it's with deep pleasure that we welcome you all.
Great pleasure that I introduce the first speaker, Dr. Eric Schoen, who's Professor of Genetics and Development at Columbia University. Eric and I go back a long way. We've been collaborators in very interesting projects in the past. We were involved a little bit in the cloning of Dolly. And he's fearless and courageous in going to areas that other people have tried to venture and tried to resolve. But I think that Eric is one of the more creative and innovative scientists that I know. And you should know that he's doing really pioneering work in Alzheimer's disease. So it's really appropriate that he's speaking, even though on a different topic, it's speaking in this Neuroscience Institute. Eric is also a great teacher, and one of the few people who I know who in 45 minutes is going to teach you everything you want to know about genetics. So after you listen to his lecture, apply for a PhD, and you'll be in good shape. So well, let's welcome Dr. Eric Schoen.
Good morning, and I want to thank John for inviting me and for the organizers. With that kind of an introduction, I don't think you need the 45 minutes. You can get the PhD right now. Okay, so you can all hear me. So my task, as I see it, is to be the appetizer for the next 2 days, to set the table for what is to follow. An underlying scientific basis for some of the things you're going to hear. I obviously cannot cover everything, but we'll try most of those things. And I want to divide the talk first into some basic concepts of biology, which many of you know, but maybe some in the audience don't. Then I'll talk a little bit about technology, and we'll end with actually an interesting problem that has arisen in the field of my own field, which I'll tell you about in about 30 minutes. So why don't we start at the beginning with a cell. And all I want you to know about the cell right now is that, besides being the source of genetics, there are two places in the cell where DNA resides, and DNA being the basic information in the cell, obviously it resides in the nucleus. It resides in one more place, which is the mitochondria, and I will tell you about that later. All the rest is commentary. Fundamental thinking in our field is Watson and Crick's famous central dogma of molecular biology. Almost everybody knows this. DNA, which is a stable source of information in the cell, encodes RNA. This is deoxyribonucleic acid, encodes ribonucleic acid, which is a transient source of information in the cell, which then, and is movable. This is immovable, this is movable, and eventually encodes the actual business end of what cells do, which is fundamentally proteins, and via proteins, carbohydrates, fats, and everything else the cell does. This is an iconic figure. You don't hear me. Sorry. Better? It's an iconic figure. The double helix has now entered society everywhere. Just to reiterate what the double helix is, it is merely a backbone, fundamentally of sugar and phosphate, two backbones that intertwine and are connected by connectors, which are called the bases, and there are 4 of them. There are actually 5, but the 4 here are guanine and cytosine, G and C, thymine and adenine, A and T. G always goes with C. A always connects to T. You can never have it any other way in a normal situation. That's important because that provides the fundamental base pairing rule, which was the basic insight of Watson and Crick in terms of DNA replication. So this is what it actually looks like in a microscope, at least a little bit of it. You can barely see the double helix, but there it is. And that's what it really looks like when somebody in my laboratory isolates it. You can spool it out of a beaker, and it looks like, oh, this is what it looks like. Okay, do something. Try again. Here we go. So that's the DNA as you see it in a cartoon. Most people think of DNA in chromosomes that look like this, but actually that's not what it looks like inside the nucleus. Here's the DNA, and there's a huge amount of it. 3 billion base pairs in the germline. That means what's called the haploid genome, a single set of chromosomes. In every other cell in the body, 6 billion base pairs, twice as many, half from the mother, half from the father. And it's all wrapped up, and I'll show you this a little bit more, and it looks like a bowl of spaghetti inside the nucleus. It does not look like this, except under special circumstance. But that's the circumstance that is easiest to spot, and the circumstance we know best. And the reason for that is because how do you put 2 meters, 6 billion base pairs is 2 meters, that stretches from here to there, into a cell that is only a few hundred microns in size, a few hundred micrometers. So it's spooled up like on a fishing rod. And you can pack an enormous amount of material in a linear version, in 1 dimension, and put it into 3 dimensions. So this is 2 nanometers. That's 2 billionths of a meter. And then it's spooled around, just like this one. It's spooled around proteins that are called histones. And then those wrap among themselves, like this. And then those wrap among themselves, like this. And you can see we're getting larger and larger. And then this is just a little segment here, okay? And that becomes a segment that's wrapped around again. You are decreasing the volume of the cell, of the DNA, by a factor of 10,000 when you do this. I mean, it's an amazement to see this happen. And that's what they really look like. These are called metaphase chromosomes. And they're actually, each chromosome, and the beautiful pictures here of this, each chromosome is actually a duplicate of its sister. These are 2 sisters that are absolutely identical and are connected in the middle. But remember, when DNA is actually being operated on, it's a single long spaghetti. And there are 23 of them. And here's the first piece of new technology. This is called chromosome painting. You can actually identify each chromosome using special kinds of fluorogenic probes, and which you can see distinguishes one from the other. In the microscope, you can do this. So, DNA and the central dogma, the first part of the central dogma is that DNA must be replicated. The base pairing rules, G always goes to C, A always goes to T, means that if I can take the double helix and unwind it, and I have GTC on this side, and I have a machinery in the cell to create a daughter strand, which would be the red strand, which is being created here by special enzymes, that the next letter here will have to be a G. The next letter here will have to be an A. The next letter here will have to be a C. And in fact, therefore, you will be able to use the information of one strand to derive the information on the second strand. So from one strand, I can make two. And by the way, if I did that here again, I could make four. But we only make two in the cell. That's all you need to know about DNA replication. It's stable, it stays inside the nucleus, and never goes anywhere else. However, the cell is much bigger and needs information that is outside the nucleus, and that's created by the RNA. So here's the double strands of the DNA. We separate out the strands, and there's an enzyme that does that, and reads one of the strands. So here we have ATC, GCG, and so forth. And from that, special enzyme creates, just as if it were a complementary template of DNA, this time it's a complementary template of RNA, ribonucleic acid. So now C makes G, G makes C. A does not make T, it makes a related base called U, or uracil. That's the fifth base, by the way. And so now, if you read off the green strand, you make the orange strand. Why? So the first why answer to that is that this piece of RNA is mobile. It can actually escape the nucleus and go into the cytoplasm. The second answer is that DNA is very stable, very resistant to stress. RNA is very unstable and can be degraded rapidly. So the cell has a mechanism to transfer information from one part of the cell to the other, turn it over, and then make something else. And in long-lived cells, the RNA may be long-lived. In short-lived cells, it might be short-lived, or vice versa. But it's a way to have a dynamic control over information inside the cell. So once you've made from the DNA, you've transcribed the RNA, just like a secretary transcribes. Each three letters here, so from the ATG now we have AUG, CGG, and so forth. In a frame of 3 by 3 by 3, each triplet, which is called a codon, will encode a protein. And a protein is made out of 20 amino acids, one of 20 amino acids. I'm showing you 4 of them here. And so as you go along this way, you're encoding a physical structure called a protein, which starts with thymine, arginine, tyrosine, leucine. I won't give you the other 16. How does it do that? It uses the famous genetic code. So the genetic code is a way to convert those 3 letters into the information of the proteins. Since you have a triplet code, and each position in the code, A, T, G, the first position could be any of 4 letters, A, T, G, or C. The second one could be any of 4. The third one could be any of 4. 4 times 4 times 4. There are 64 possible triplets. And of the 64, 61 of them are used to encode amino acids. 3 of them are encoding nothing. That ends the string of letters. And so before I had methionine, arginine, tyrosine, leucine on the previous slide. And here they are, AUG, CGG, UAC, UUA. That's how the genetic code works. And it's done by an incredible machine for which a Nobel Prize was given out last year, and Israel won 1/3 of that prize up at the Technion. Here's the message. This is an adapter called a transfer RNA. Here are those 3 letters, let's say ATG. So the mRNA is attached to the adapter, and out of the end of the adapter of the ribosome, which is what this big machine is, and for which the Nobel Prize was given, out come the amino acids. And the amino acids encode a protein. And here's another way of looking at it. They're actually, these ribosomes actually run, sometimes you have multiple ones, and as it's running this way, out is being spewed the protein. And you can actually see that here. These are the ribosomes. There's a real photograph, an electron micrograph, and the proteins are coming out. And as you can see, as you go from here to here, the protein's getting longer and longer. This is the way it actually happens in the cytoplasm. Now, let me drop back to RNA for 1 minute. What I didn't tell you that is in humans, the gene encoding the RNA is actually in pieces. So this is a part that's going to encode a part of the protein, and here's another part. These are called exons, and another part, and another part. And the green material in the middle is fundamentally non-coding information. So when the gene is transcribed into a messenger RNA, and that's what it is, a message, it actually creates a long precursor RNA, and then the white boxes are thrown away and are edited out. So you go from 1 to 2 to 3 to 4 to throw away the middle stuff. But sometimes you can make 2 different versions, 1 and 2 and 3, here to here to here, skip this, or 1 and 2 and 4, here to here, skip 3, go to 4, or you can make 1, 2, 3, 4. Right away, one gene here can encode 3 proteins. 1, 2, 3, 4, 1, 2, 4, 1, 2, 3. You can see, therefore, that even though there were 20,000 genes in the human genome, you could have 100,000 proteins, 500,000 proteins. And that creates some of the diversity in the mammalian genome that makes us special. Finally, what's a protein? I could show you hundreds of structures of proteins. This is one that a friend of mine crystallized and identified. So this is called thyroidox. It's not important what it is. Just to tell you that as the message spews out, it spews out from one end and will go to the other end, just like this, down to here, up to here, around, up, around, down, up, and down, and out. And it folds in its own unique three-dimensional structure. And it's that structure that creates the protein that does what the cell wants it to do. Any disruption in this section, in anywhere, can create a problem. You can maybe appreciate that if I have different exons, 1, 2, and 4, they can actually create modules. Maybe exon 1 is creating this part, exon 2, this part, exon 3, this part, exon 4, this part. What if I go 1, 2, 4, I do this and this and this, but not this? Sometimes that's good, sometimes that's not so good. But nature has evolved to create many flavors of proteins out of a minimal amount of information. All right, that's all we need to know about biology, the normal biology. So let's talk about when things go wrong, because we're going to talk about the next 2 days when things go wrong. So let's start with the big picture. Large-scale chromosome rearrangements of some sort. Now, I'm only going to talk about 2 of them. The biggest large-scale chromosome rearrangement one can think of is just getting rid of a chromosome or having too much of a chromosome. And the one I'm picking is called non-disjunction and is actually an example of which Down syndrome is an example. So in the germline, that means in the oocytes of the mother, in the sperm of the father, cells are actually haploid. That means they only have one of the 2 parental chromosomes. But before that happens, all right, there has to be a duplication of chromosomal material, and that's what this that's the classic picture of the metaphase chromosome I showed you. One of these will divide and go into the germline, and eventually, at fertilization, this one will go here, and this one will go here. Similarly for the mother, this one will go here, and this one will go here. So this is a normal fertilized egg, 2 chromosomes. One from the mother, one from the father. And here's another one. In non-disjunction, which is a terrible word, okay, because it's a double negative, these 2 don't divide. They don't come apart. These come apart, but these don't. So this one goes here, this one goes here at fertilization. Excuse me. Right. But here, they didn't separate. So both of them wind up here, and nothing winds up here. This cell is completely unviable and will die. In a few cases, for 3 different chromosomes, of which chromosome 21 is the example. You can have 2 chromosomes from one parent, 1 from the other, meaning a trisomy. And for trisomy 21, the child is viable. That child will have Down syndrome. Trisomy 18, other genetic problems. Most trisomies are not viable. Chromosome 21 is. And that's a classic example of a genetic problem. We have no idea why this problem actually increases merely with the age of the mother. Once a mother passes the age of 35 or 40, the probability of having a child with a non-disjunction event climbs exponentially, and we don't know why. It's an active area of research. Now, I told you that in the germline, we're separating chromosomes. But before that happens, the chromosomes, in order to have cell division in the germline, you have to double up chromosomes, and eventually we're going to have them later. But after the doubling up, you can have crossing over between maternal and paternal chromosomes. And this is normal, in fact, is necessary. And it is the reason when you get this shuffling of similar information. Let me be clear. This chromosome, let's say, is chromosome 5. This is also chromosome 5. So these are fundamentally identical chromosomes with similarity all across the way with minor variation. And because they are so similar, because of the base pairing rules, you could have a piece of the orange chromosome actually crossing over and aligning with a piece of the gray chromosome. And when that happens, you get a crossover event such that some of chromosome 5 from the father now winds up on the mother, and reciprocally, some of mother's 5 winds up on the father's. So when you then go into division and make the new baby, the baby looks almost exactly like the parents, but not exactly. If this didn't happen, we would all be clones of each other. But we're not, because this generates the variation that makes me look like my mother and father, but I don't look like my mother and father. That's outside and inside. How does that happen? It's a wonderful process. This is actually a cartoon of a crossover junction. It's called a holiday junction. 2 L's. It's not the holiday. It's Robin Holiday who discovered this. And here's a beautiful picture of a holiday junction before resolution. And then you cut, and then you can see that you can bring together 2 different dissimilar pieces, maternal and paternal, together on the same chromosome in a reciprocal way. So, because we know this now, besides having extra chromosomes or fewer chromosomes, you can now begin to exchange chromosomes. I told you that recombination occurs between homologous chromosomes, maternal chromosome 5, paternal chromosome 5. But in fact, there are areas on every chromosome that have regions of similarity or homology one to the other. So here on chromosome A, let's call that chromosome 2. And here on chromosome B, let's call that chromosome 11. There might be a small region right here where there's a long enough region of similarity that just by accident, instead of crossing over with its homologous pair, it crosses over with a non-homologous pair, meaning A crossing over to B. And now you've brought a piece of the orange to here and a piece of the purple to here. All right? This is not good. You don't want that. Sometimes it happens and it doesn't matter because only 1% or less of the genome actually encodes proteins. 99% does not. So if the breakpoint happens, if you will, in a gene desert, probably not that important. But if it happens in a gene, big time problem. And one of the classic causes of cancer is exactly this. It's actually called the Philadelphia chromosome, from where it was discovered. What is more normal is a micro crossover, not between chromosomes, but inside of chromosomes. Just as there are regions of similarity from one chromosome to the next, there are also regions of similarity inside the chromosome itself. And so then you can have what's called a copy number variation. So normally, here's a region of DNA on the maternal and paternal chromosomes, all right? This region of DNA might have a region of similarity somewhere else, such that the junction, that holiday junction, it will be a folding over. And now you will duplicate a region of the genome. This and this are now identical. Or the other way around. You can actually lose this information. And now there'll be a missing part here. This is called copy number variation. As with the crossing over the big orange and purple chromosomes, if the variation is in a gene desert, probably not important. If the variation is within a gene somewhere, it probably is important. But because this is normally homologous to this, if this is not a gene, this isn't a gene. And therefore, the intra-chromosomal copy number variation is probably not that bad.